right, Dan, you ready to get rolling?
Yes, I am.
All right, and welcome everybody again for Data Science for Biomedical Professionals. We're happy to keep this conversation rolling today. Again, make sure you've pinned Dan Chan's video so everything stays in the forefront of your screens. But welcome back. For anyone that might not remember, we have all of this information and we're going to be posting and sending an email out with the recordings. We're finishing the render of Tuesday's session. We cleaned that up just a little bit, so it's going to be nice and neat for you all to reference that at any point in time. We also have pre-survey and post-survey workshops, which we would encourage you all to take if you're interested, even just for giving feedback, not just participating in the research study, because we would really like to make this a useful set of workshop series for now and in the future. And just as a heads up, we'll be talking about this more towards the end of today's session, but we will have additional workshops that follow on to these that'll keep going into more and more topics in R and the analytics side and visualization side of that. So be on the lookout for us to be promoting those. And we'll obviously also be offering these again in the future. So we have that information. I'm Dr. Brown. It's nice to see you all here again. I think we've got everyone from Tuesday, so I won't go into depth of who I am, but I'm here. Please feel free to message me in the chat individually or chat into the group for questions, and we'll help you along the way. We also have two helpers with us, Jonathan Berganti, who's a lecturer in university libraries, and Carter Gottschalk, who's a research assistant. We're all here to keep you learning R and feeling confident about it. We have a couple other resources for you as well, which you see down here in the bottom of the screen and in the chat. We're going to have an Etherpad, which is like a live notes version of what's going on today as well. So you're going to have this both as an audio recording and you're going to have a written notes section as well. And all of our lesson materials we're compiling on our website, ds4biomed.tech, and they're accessible at all times. With that, I'm going to hand it over to the mastermind behind this, Dan, and he's going to get rolling with our work today with tidy data and analysis and visualization in R.
All right. Hello, everyone. Thank you for coming back for the second day of our series. I introduced myself on the first day, so we won't get too much into that. But just so you see all the links on the screen, the actual book version of everything I'm talking about is on ds4biomed.tech. There is a link to the slide decks if you want to see it. And a few of the papers that I will be referencing, there's also the PDFs for them in that Google Drive as well. As far as the slides go, they are literally screenshots out of the book version. So just so you know that everything is actually written in the book version of our current materials. The Etherpad is a really nice system. Carter is actually manning that right now, taking the actual notes from the class, but feel free to also go on the Etherpad and add your own notes in there as well. And if you don't want to ask a question through the Zoom chat, either through a DM or anything, you can post a question in the Etherpad. So right now we are all the way at the bottom at line 133 is where today's Etherpad starts. And then when this is done, this will also all be posted online. So you will have a running log of all of the notes. And then last but not least, just moving forward, there is a big glossary project being maintained by the Carpentries. So if you run into any technical terms, this would be a good place to look if you're just trying to get a definition of something. Last but not least, this is an example screen layout that was suggested from Tuesday. This is really made for if you have a single monitor screen and you're following along today's workshop. The main takeaway is think of your screen as 4 quadrants. Have my current video in one quadrant, have RStudio open in another quadrant, and then you have the Zoom stuff in another one, and then the last one would be something like the Etherpad or like a Google another tab for you to do other things, and then just resize them vertically as needed. And with that, we will one more topic. If you do end up going to the live ds4biomed page, and occasionally you end up seeing this message that says page not found, but it worked like maybe 10 minutes ago. I am currently in the process of fixing this, but this is what happens every time I make an update to the website. It actually re-executes all of the code from scratch and installs all of the packages from scratch. So it's sort of a guarantee that I know that everything that's presented works. The downside is it takes about 15 minutes to do all of that, and in which case, during those 15 minutes, the site goes offline. So these couple of days, I tried to do all of these site updates like around midnight. So hopefully no one's like super actively like reading this stuff. But just so you know, if you do see this page not found error and it worked before, it's most likely a site update going on. Okay, so the first topic for today is going to be on this topic called tidy data. And if you took any of the surveys, I need to go to the beginning. How do I do that? There we go. There was a very specific question asking if you knew what this was. And so in this first part of today's class is we will thoroughly cover what this term means and what this means when you're processing your data. And so this is really the first step when we are talking about data wrangling or cleaning your data set. And when you're putting together your own data set, this is sort of what you want to keep in mind as you're creating your own data set. So Tuesday's first class, when we went through all the spreadsheets, I went through a bunch of examples of like setting up your own data. When I was talking about like, think about the future, how you might use this data set, this is that future part that we're talking about. And so what we will be going over today is actually almost like a journal club paper review of this paper by Hadley Wickham, written in 2014, titled Tidy Data. And essentially this is essentially why this paper is really great is because it sort of starts to formalize what it means to have a clean data set, right? So we always talk about like, oh, my data is always messed up, and we don't know exactly like why it's messed up. This is the paper that actually gives you like written down in words that you can understand, like how you can describe a messed up data set. And so these are all screenshots from the paper, but we'll go through everything one part at a time. So let's say we have this data set. Each row represents a person. So we have John Smith, Jane Doe, Mary Johnson. And then there's a column for treatment A and treatment B for a example experiment that happens, right? This isn't a real data set. And you can see this data set that will serve a purpose. Let's say you are collecting data and you're sort of like the data entry person for like a trial or something. This is a really good way for you to like make sure like, okay, I have a person named John Smith, and this reading was this, and this other reading was that, right? This is really good for, you can visually check, or you can present this table to someone else and they'll have a general sense of what's going on, right? So it seems right now, if we take a very quick glance over this, it looks like the values in treatment B are lower than the values in treatment A. And this is a visual thing that we can do. We can transpose this table, meaning that we can flip the rows and columns, and we get a very similar representation, right? None of the data, so the most important thing is the actual cells in the data set don't change at all. This is just a different representation of our data set. And in here, we can see like, okay, we can still do that visual check of treatment A and treatment B. You can see like it is literally like wider, meaning that there are more columns. And you can see like this can be a sort of an issue of trying to see A and B when, we have more people. Right now we have to scroll to the right instead of scrolling up and down. And because there's only 2 treatment values, like if we have like 20 people, it's going to be a little bit visually difficult to like compare treatment A and treatment B. But if your goal is just to make sure that you have a value for each person, this still does serve its purpose. You can quickly look down. Even in a previous example, do you have a value for each person? That's a really quick way to make sure that during the data entry process, this all works. So the last thing we talked about on Tuesday was like that group by calculation, right? When we said like, hey, I wanted for every different type of treatment, give me the average something, right? So when we talk about tidy data, this is actually the form that Hadley describes as being tidy. So at first glance, this actually, like, from a person reading like a table, looking at a table, this is actually harder. This is actually like the most complicated way to make sure that, do you have a value for every person? Well, then you might have to sort by the name to make sure and then count, right? Can you do a quick check of which one's like lower or higher? It also becomes harder to check. But from a computer's point of view, we can now say group by treatment and then give me the average result, just like what we did in our last bit on Tuesday, right? So we can actually tell the computer, like, split our data set up by each different value of treatment, so A and B, and then take the result value from each of those pieces and then average them and put them together. So remember, group by is splitting our data set into some set of columns, applying a function, in which case the mean is one example, and then putting it all together so we get a table back. And then we can also even ask the question of if we want to fit a model, we want to see like, how does treatment affect the result? We can now say pick the treatment column, pick the result column, and those are our two columns for our model, right? And so you can see that there is one format that's useful as like a reader or as a table representation, and then there's another format that's more useful from a computer, from an analysis perspective. And that's the whole, what's the word I'm looking for? That relationship is what tidy data is. So our end goal is to get our data into this format, and then we can very easily convert it to any of the other formats. And in the process of getting it into this tidy format, there are very common data problems that we can all fix. And so what do we mean when we say tidy data? What makes a data set clean? There's three parts. Each variable forms a column. So we have a name column, a treatment column, and a value column. Each observation forms a row. So what is the unit of observation that we care about? It is the person treatment value. That's what we care about. So each of those will create a row, and then each cell is a single measurement. So remember, we're not putting too much information into a single cell. So we're not going to use colors to represent a bad value. We'll have another column to tell us that a value is bad, right? We don't, for any given number, I shouldn't be able to say like this thing encodes two different things, right? So like if I have a number for temperature, that could be a temperature value, but like, let's say you're recording your temperature in Celsius, Fahrenheit, and Kelvin for some reason, that you want a separate column for like the actual unit, for example. So each cell should only represent one thing. And so what this means is data can be messed up in a whole sorts of ways, but we can use a common set of tools to process them and make them all tidy or clean in the same manner. And so once we have our data in a tidy form, we can then convert it into any other form. Another good thing about having tidy data is once you have your data set that's tidy, you can actually combine it with other data sets. So it's not always going to be the case where you are working with a single data set. Working with demographic data is very common. So you have, your patient population in your clinic or in your lab, and then you want to put in or start grabbing in the overall demographic data from census. Well, you need both tables to be tidy so you can combine them together. And so this gives a formal definition to clean our data set across all of the different data sets that we have so we can work with them later. And so what are the common data problems that we have? So we will cover each one of these one at a time. So one common problem is you having columns your columns are values and your rows are variables. You can have variables in columns and rows, or you can have multiple variables in a single column, and et cetera, et cetera. So we will go through each one of those points. So back to the actual tidy data paper. The first common data problem that you will have is column headers are values, not variable names. So what does that mean? This is the example table that Hadley has in his paper. It comes from the Pew Research Center, and it is the relationship between religion and income, and the body of the table represents a frequency count. And you can see this is a very dense table. We can actually present a lot of information in the single slide. But if our question is, you know, for every income bracket, what is the total number of people? Or if our question is like, for every religion, what is the total number of people? We can't answer that from ours perspective. And you want to get out of the habit of, yes, in Excel, what I can do is at the bottom of that row, create like equal sum and then sum the column. And then you want to get out of the habit of that, because when you do that, you actually just entered an actual piece of data in your raw data set. And so you can either accidentally forget to delete it when you want to process it later on, or you add more rows and then you forget to drag the summing columns, and you actually end up missing bits. So what we want at the end is actually a data set that looks like this. Again, the problem is this is a very long table, and it's not a very efficient table in terms of representing it on the screen. But from a computer's perspective, we can now say, how many people do I have total? I can just sum up that frequency column. How many people do I have for just the income bracket that is 40 to 50K? Well, I can filter and then sum the frequency column, or I can do a group by aggregate. I can even say a bar plot of like, hey, use the income value as like the X, and then sum up the frequency and use that as the bar height, right? So the takeaway is you can specify a single column to do those operations. Yes, okay. So we are going to load up R, and we are going to go through a series of examples. So back into RStudio. Make sure that we, at the top right corner, are working in the RStudio project that we worked on before. And so make sure that the top right corner should say something like ds4biomed -learner. I believe that's what it should be called if you downloaded it from Tuesday. What it should do is in the bottom right hand corner, you should see the two files that we created on that first day. So just give me a green check mark for yes if you can see everything on my screen that I'm presenting. Okay, cool. All right, so we are going to click on the top left corner, go to, you can either go to File, New File, RScript, or you can click this little white paper with a plus mark and click on RScript. And that should open us up into a new script file. I'm going to save this file, and I'm going to label this 03 tidy. And so again, remember, I'm using a numbering system, so you can see like the order that I'm showing you these lessons are going to be sorted in the order that they're presented in. Okay, so the first thing that we have to do, this is a new script. We want to load up our tidyverse so we can work on loading up a data set. So we can say library tidyverse and hit Control Enter, and it should load up the tidyverse packages at the bottom. Next, just like we did on Tuesday, we're going to load up a data set. In this case, we're going to load a data set called tumor growth. This is a data set that comes from Peter Higgins, who's a medical doctor in the IBD research group at the University of Michigan Medical School. And he actually created a R package called Medical Data that has medical data sets that we can use. What we will work on installing that package after this part. But this is a data set, and I'll sort of just read the code book from it. These are cells from human glial cell line were implanted in the flank of N=37 nude mice, and a subcutaneous tumor or a xenograft was allowed to grow. When a tumor growth grew to around 40 to 60 millimeters cubed, the animal was assigned to one of 4 experimental groups. And then the main outcome in xenograft experiments is the volume of the tumor over time. So we will be working with a tumor data set that has the main value of interest is the tumor growth size, and these are how the size of the tumor over time. So let's look at this data set. We're going to save it to a variable called tumor, so TUMOR. We're going to read _CSV to read our CSV file. And then in double quotes, we are going to say it's going to be in the data folder, and then the tumor growth _long.csv. I sort of made a mistake in calling it long when I created the data set. So that's sort of an artifact of after I was cleaning his data set for teaching purposes. So if you were to come back and re-download the data set, I believe like a month from now, like the name of the files will be a little bit different, but the names on the actual website will be up to date. So once we have this, you can see that the top right corner, this tumor page variable loaded up, and we can explore this data set. So we can see we have a column called group, a definition of the group like control, CTR, D, etc., an actual ID number. And then we have columns labeled 0 to 28, and then the body is a number that represents the size of the tumor in millimeters cube, right? And so if we look at this data set, we have that same problem from the Pew Research data that I showed you from the Tidy Data paper. We have columns 0, 1, 2, 3, 4, 5, 6, 7, 8. Those are actually values of a variable and not an actual variable itself, right? So that 0, 1, 2, 3 represent days since inoculation. So we should have a column that's like days, right? We shouldn't have day 0, day 1, day 2, et cetera, et cetera. So when you're working with your data set on your own, this is something that when you're creating your own data set, this is something you want to think about. There's nothing wrong with inputting data this way, but just know that in the data cleaning process, something needs to happen to this data set to make it a little bit easier to do analysis with. When you get a brand new data set that you've never seen before and you see something like, wait, these columns actually represent a overall variable. These are all the little symptoms of our data that you want to think about on how to clean it. And so how do we actually process this data? We are literally taking it from like a data set with a lot of columns, which is like another way to say it is it's a very wide data set, and we want it in long format. So there's more rows than columns. So the way we do this, since we're going from wide to long, there is a function called pivot longer, and that's the function from the tidyverse that we will use to clean this. So the way we can do this is tumor gets passed into, so remember the pipe operator is % greater than %, or hold shift, and then it's 5.5. The function that we're going to use is called pivot _longer. And the way pivot longer works is the first argument in pivot longer, we have to select the columns that we want pivoted longer, right? So if we look at our data set right now, we have group all spelled out, then GRP, and then ID. Those are the columns that we don't want to touch. And then all the other columns, those are the columns that we want pivoted in longer format. So we need to specify all of the columns from 0 to 28. And so on Tuesday, I talked about, you can use the select function to select the columns that you need. But in our current case, it is not exactly, it's feasible and you can do it, but you don't want to have to specify 0, 1, 2, 3, 4, all the way to 28. And so the tidyverse gives us a bunch of extra ways for us to select columns. And so I'll show you one of them right now. One other thing that you'll notice is when we loaded this data set into R, because the column B started with a number, it was put into a series of backticks. So a backtick is the key to the left of the number 1 on your keyboard. This is not an apostrophe. And so in general, in terms of when you're naming your columns, have it start with a letter and not a number. That's sort of one of the rules when you're working with data sets. Otherwise, you have to end up typing more characters to make this happen. So if you were to end up doing this on your own, you might have a column that's like D0 or D1, et cetera, et cetera. Okay, so what we want to do is select all of the columns from 0 all the way to 28. So how do we do that? We can first specify the 0 column, so backtick 0. And then the way we can select everything is we can put colon and then backtick 28. And this is the tidyverse way of saying, I am selecting all of the columns from the column 0 all the way to column 28, right? And that's one way to do it. Since I know 28 is going to be the last column in our data set, just in case there's a 29, 30, 31, 32, you never sort of want to get out of the habit of like putting in hard numbers. There's an actual shorthand for last underscore column as a function. And then what this will do is I'll take the column that is labeled 0 and go all the way to the last column. So those are two different ways of selecting the column. The next thing in pivot longer is we're now taking those columns and we are making them into long format, right? So those columns that were labeled 0, 1, 2, 3, 4, all the way to 28, that's going to turn into a single column. And so now you want to specify what is that column's name going to be. And we specify that with the names underscore 2 parameter. So we can say names2 is equal to the word like day, because that's what it represents. So when it does that pivot, we have a column called day, and then we have like the values down here that we have to deal with when the table's getting flipped around. And so we need another name of a column. And so what represents these values? We are going to say values underscore 2 is going to be a column called size. And now if we run this, sorry, it's not lost column, it is last column. So I spelled that wrong. If we run this, we actually get a long form data set from our tumor growth. And so if we want to work with this later on, we can come back and say tumor underscore tidy gets this value. And so now we have the original tumor data set that we loaded in, and then the tidy format. And you can see it's in longer format. There's fewer columns, so 5 variables, and now 1,000 rows. And so what actually ended up happening here? So here is the graphical representation of what is going on. So we had columns X, Y, and Z, and an ID column. So those were our group and ID column. And when we ran pivot longer, we had the value of X had the value of A and B. So now we have value of X had a value of A and B, and then Y became C and D, and Z's value became E and F. So the pivot longer transformation that we just did, where we said start from X to the last column or select all of the columns from X to Z, this is the transformation that happened. And if you want the GIF version of this, you can see that going from Y data to long data is using that pivot longer function, and they're related to one another. And so I'll show you at the very end. how we can undo the pivot longer page. Give me 1 second. Okay. So now that we have our tidied data set, we can actually then ask another question. So back to the last thing that we did on Tuesday, what happens if we sort of say group underscore by, and we're grouping by the group column, so with the capital G, and then we're also grouping by day. We can then say summarize, our average size is going to be the mean of the size column, right? So what this will do is give us the average size for each day for each of our groups, right? And so if we run this, you'll see that we get a bunch of NA values, right? Meaning that it's not the case in our data set that every single value for like group 1 day 15 is missing. We can come to our original data set and we can see like for 15, like we literally have some values. And so the way R works is if you give it a lot of these summary statistics, if you pass in a bunch of values and one of those values has a missing value, it'll render the entire calculation as missing. So the way we fix that is in the mean function, there's a parameter called na.rm that stands for na is missing value. Remove this missing value and set it equal to the word true in all caps, right? So I will paste this in the actual chat for everyone to see. And so now if we run this, if the calculation for mean encounters a missing value, it will not count the missing value. What it's not doing is treating the missing value as a 0 and putting a 0 in your average. It is not treating that number in part of the denominator of the calculation at all. And so now we can look at our data set and say for every group and every day, we can see the average size of our data set. And we can we're only able to do this because our data sets is in our tidy format. One other thing that you'll see is under day, it is actually being treated as a CHR character value. And what we want to be able to do is turn the day column into an actual numeric column. And I will paste in the chat for everyone the pivot longer command. And so given this data set, there is another tidyverse function called mutate, which allows us to modify a column. So in tidyverse, we learned that select is how we can select columns, filter is how we can filter rows. We learned about group by, and then mutate is how we can change the value of another column. We can either change it in place or create another column entirely. So we can say mutate, and we're going to overwrite the day column and turn it into a number. And how do we turn values from one data type to another? There is a function called as.numeric. So if we type in as dot and you sort of wait a bit or hit tab, you'll see that R has a bunch of different ways to convert types. So you can turn it into a character, so it's CHR. You can turn things into a data frame if that's what you want. You can turn things into a date object. And so the current thing that we're working on is turning it into a number. So we can say as.numeric. and then we pass in the column day. We're going to turn it into a number and then reassign it back. And so now if I run this, we can actually see that our day column is now a double or a number. And what that means is now because it's a number, we can sort this properly, right? Because we have 0, 1, 10, 11, 12, not 2, 3, 4, right? So now we can actually sort our values correctly. And the way we can sort our columns is using a function called arrange. So we can arrange our columns, and we now can pick what is the order that you want to sort our values in. Maybe you want to sort by the value for group first and then day. And then so you can run this and you'll see that now everything is sorted in correct numerical order. Maybe you want to sort the day in reverse, in which case you can say minus day, and it'll sort the days in reverse. And you can see for group 1, there's actually no values for anyone from days 22 to 28. Maybe you don't care about the group and you only care about it being sorted by day, in which case it'll see all of the 0 days first, then all of the 1 days, and then the 2 days, etc., etc. So mutate is, we're only able to do that because we turn the day column into a number using as.numeric, and then we can specify different ways of sorting. So I'll put that back. And if that's what you want to do for your little analysis, and I'll paste this into the chat, then this is how you can start running summary statistics. Okay. Okay, so give me a green check mark if you're okay up to this point and I'm able to move on. I know that there are some people that were having technical issues, and so let's just make sure that everyone's sort of caught up to our current location. Or if you need help, put a red X, and then someone can come message you, or I can talk you through how to fix something.
Hey, Dan,
Okay.
we had one question that I'm not sure if we got it earlier or not, from Shaga. She's asked if can't we just transpose the data when we were still working in pivot longer areas? Any comments on that?
Transpose the data. Oh, so you mean, okay, so let me see if I understand the question correctly. Given the tumor data set that I have now, your question is, can we pivot these columns, or sorry, transpose those columns? So the transpose function, all it is doing is swapping the columns and rows, right? So if I go back to the slide. So if we have our original slide here, table here, transposing is literally turning treatment A to a row and then this column here. So we're literally just swapping the tables, right? So we go from a table with three rows and 2 columns to a table with 2 columns and 3 rows, right? So transposing is literally just swapping those 2 values. When we're doing pivot longer, it's actually a totally different operation because we don't have 3 and 2, we have 3 columns and 6 rows, right? And if we go to the actual comparison between the relationship between wide and long data, it is more than swapping the rows and columns. So the question is, if it's wide data, doesn't it help if the rows are not many? I don't know what that question means. I guess like if the question is around like, why go through this process? Because I can sum up the 0 column, and I can say like group by group and 0. Is that sort of what you're asking? Yeah. So when we get to this point here, when we're trying to calculate a aggregate statistic, if it wasn't in this tidy format, we wouldn't be able to pass in a value for this mean function, right? So if we said something like group by, so we could say something like group by the group variable and then give me the average of the 0 column. That's correct. If that's all you care about, then you don't need to go through this whole pivoting process. But if your question is, I want to see how the average size changes across multiple days, unless you're willing to say average size 0 equals mean of 0, average size 1 equals mean of 1, all the way to average size 28 equals mean of 28, unless you're willing to type that, it is much easier to say group by the group and day and give me the average size of that other column, right? So it not only allows you to make your code like less error prone, because like at that point you're literally just copying pasting, and so nothing's stopping you from like accidentally putting like 2 15s in there, and then you're trying to draw a table and then you have a flat peak which you might think is meaning something, but it's because you copy pasted something wrong. So the tools are there to also prevent you from copying pasting things over and over and over again, so you can make those mistakes feel less.
All right, I think that gets us into some good directions with tidy data. Let's take it to the next point, if that sounds good to everybody.
Okay, cool. And if you're running into issues about something like column does not exist, one thing that you can do, there is a function called names, and if you give it a data set, it will just print out all of the column names. So if you're getting something like you know, the 0 does not exist, make sure that you're, one, working on the correct data set, and then two, hopefully the data set that you're working in has that column. So it's all different ways of checking to make sure you are in the correct spot, or you think you are where you think you are where you actually think you are. All right. So the next common data problem that you'll see is multiple variables stored in one column. And so this comes from, again, the tidy data paper. This is a screenshot from there. And this is the WHO tuberculosis case counts for across different countries and across different years. And again, this is a very dense table. We can see like we have almost all of our male age groups presented, and then double the width for the female age group. And that was cut off just for so it can fit on the screen. And so the first thing that we notice is our columns have some symptom of the previous example that we have, right? This M014 stands for males 0 to 14, males 15 to 24, up until males unknown, and then female 0 to 14, et cetera, right? So just by looking at the column names, you typically don't have a column called male and a column called female in your data set, right? Because for any one person, for every one unit, like you wouldn't have a value for both of them that's like true, right? In this particular data set, or in a particular age group, you're not going to be 15 to 24 age group, and then also 25 to 34 age group, right? That age doesn't work that way. So you can see this has that same problem as before, in that there should be a column called gender, and there should be a different column called age group, right? But we have it in this really useful condensed view, but it is sort of messing up our data set in some way, right? So we can't actually say, like, give me all of the counts for people aged 35 to 44. How many people do I have in the world for a particular year? You can't really answer that question without pulling out two totally separate columns in your data set, right? So yes, you can, in theory, do all of these tasks. If you're in Excel world and that's what you're used to, you can sum these columns, but that's sort of more work because you have to like specify these two totally separate columns, right? And so how would we clean up this data set? Well, we first do the same process as before, which was that pivot longer. So we get the version on our left. And so we end up with a column that's M014 and then that case count, right? So that solves things part of the way. So we can at least now say, like, for each group, for each year and country in the world, like, how many males 35 to 44 case counts we have, right? So we can go back to that last example and we can do that similar calculation. But we still have that problem of this one or this two represents two things at the same time because of this one column. And so we want actually the column on the right, which has split a column into male and a column into a column into sex and a column into age, right? And so if I were to, like, completely not having to do with any code, if I were to ask you, if I have a column that says M014 and I want you to convert it to a column that's M and a column called 0 to 14, don't worry about the dashes, how would you do it? And one way would be, hey, if we just take the first letter out of this column and treat it as the sex column, that would help parse out these two tables. Right, so we will go through this data set in R. And so we will now load up a different data set. We will load up a data set called TB, and we will say read underscore CSV. And this is going to be under the data folder, and it's going to be called TB underscore long.csv. And again, my apologies for calling it long when it's not long. I actually had to process the data set for teaching examples, and this was the long format after processing, so that's why it's named that way. Poor file name aside, what you should have on your data set in R, if you look at the TB data set, is that same table that we have from the Tidy Data paper, right? So we have a country column, a year column, M014, all the way to a column that says F65 and F unknown, right? So we have this data set, and just knowing the first thing I taught you, the first thing that we can fix in our data set is pivoting this table such that it is in long format, right? So we can now say TB, then pivot longer. And so remember, pivot longer, the first thing I need to supply is selecting columns, right? So before I showed you 0, last column, right? So there's a bunch of other ways you can select different columns, right? So one other thing that I will show you is there's a selection helper called starts underscore with. And then in here, we can give it a series of values that it will, if it begins with this, then it will select it, right? Because we want to select things that begin with M or F, we use this function called C that stands for concatenate. So this is how we can pass our like, hey, this thing that you want only takes one item, but the item that I want to give you is actually a list of multiple items, right? So think of it as like you have a bunch of eggs in a carton. I'm giving you an egg carton that contains 12 eggs, right? So I need to specify this one thing I give you, which is what the C container is. So you can also think of C as a container, but it really stands for concatenate. And inside I have the values M and then F. So it's M as a string, a comma, and then F as a string, in quotes. And it doesn't matter if it's single or double. And so what this means is I want to select all of the columns where the column starts with M or a column starts with F, right? So when you're thinking about building your own data set, these are like the small little tricks to help you later on. Especially when you work with survey data, sometimes you have a bunch of, well, in my very particular use case, like in Qualtrics, you have a bunch of random columns that Qualtrics creates, and then the actual survey questions all begin with like Q and then Q1 dot something. And if I only care about the actual question responses, I can say, give me all of the columns where the column starts with Q. And then there's a bunch of other helpers, like does it contain a value? So maybe you want the ones that say Q.1 for question 1 and not all of the other questions. So the tidyverse has a bunch of these ways to help you select columns, because at the end of the day, you don't want to have to manually type in 100 columns. If they all begin with the letter Q and that's what you care about, then you can use this helper for starts with Q, for example. We can leave out the, it is Longer. We can leave out the other parameters. Remember before I used names to and values to. If we don't supply them, it will be given a default value called names and value. And so these column names are useful when you're trying to process and clean your data set, but it will provide you a default value, and it depends on whether or not the default value means something to you or not. So this is part of the way there, right? We now have a long table. So let's save this and actually call this one TB long. Okay, so now we have our TB long table. Remember, if I were to completely, aside from code, tell you how would you separate this column into the MF column and then the 0, 14, 15 to 22 column, one way you can do it is say, break this column by the first character. And so the next function is a function called separate. We give separate the column. The first thing separate takes is the data set. So we're piping that in. So it's data then. Then we tell separate, hey, you want to split some column into a bunch of values. What is the name of that column? So the name of that column that we have right now is going to be called name. Next, you're going to split this column into, in our current case, 2 columns. So now when it does that split, give me the names of those columns that you want to split into. So there's a parameter called into, and we give it a C container or concatenation because we're trying to provide it multiple things, right? We can't, it won't make sense if we simply say column 1, comma, column 2, because you'll notice that the comma is what's used to separate between the different inputs for the function. But we want to provide one input that contains multiple things. So okay, so one of the questions was, when we selected a column starting with M and F, why is it also still showing country and year? And so we're selecting the columns M and F. Okay, so let me back up a little bit. Remember from the other day, I said there's this function called select that we use to select columns, right? And if I say the start starts with from here, this will give us all of the columns that starts with M and F, right? So that's what this part does. Because we're using pivot longer, the first parameter is selecting the columns that you want to pivot. So it's leaving all of the other columns as IDs and not touching those columns during the pivoting process. All right. So if I go back to my slide deck, we are selecting X, Y, and Z and pivoting them into longer format, but then the country and year are our two separate ID columns, and those are kept the same. And so that's the process that's going on right now. Yes, so you're not, the question is, do you select and pivot at the same time? You're not using select as the select function. We're using the pivot longer function, so that's one distinction. But the way the inputs that select takes is give me the column names and I'll filter the data set for you. Pivot longer says give me the column names that you want pivoted, and then I'll pivot those columns for you. Right, so even though we're specifying how to select the columns in the same way, the overall function that we're using is different. And so that's where the difference between select and pivot longer, they take in different inputs, even though we're selecting things the same way. Okay, back to separating. So now we have back to this TB long data set. We are separating our names column into two columns. One is going to be called sex, and the other is going to be called age_group or age. So that's how we told it, okay, you're going to take the name column, you're going to split it into two columns called age and sex and age. We haven't told it how to do that separation yet, right? In our current example, because we said just lop off the first character, separate has a convenient way to say separate on the first character, separate equals 1. And then what we can see is it'll lop off the names column, and then at the character 1 mark, that's where it will do the separation, and then you'll get the thing to the left as sex, thing to the right as age group, right? So when you're putting stuff together, you can either use a single character as a code to split things, because again, the age group, the sex age group variable, like M0 to 14, maybe that's easier for you to input into your spreadsheet, right? Like again, like all data set always ends up being quote unquote messy for different reasons because someone somewhere found that format useful. And so as long as you leave these little markers on how to convert things back, you don't have to worry about like, oh, how do I process this, right? What you don't want to do is like in Excel, like copy and paste like one sheet to another to re-pivot all of these tables, right? So that's what you don't want to do. And so with that, we can actually save this values and say tb_tidy. And now we have a clean data set for tidy, right? And so now we can go back to the aggregates and say like, oh, what are the total male and female counts for the year 2020, right? Sum up that value. And so we can start asking those questions with this format of our data set. Yeah. Okay. Last topic before we take our first break. Where's my slide deck? Okay. Last problem that we can have is, and let me just paste all of this for you. We will paste all of it. So if you're completely like behind, you can copy paste that. So the last bit is variables are stored in both rows and columns. And so in the tidy data paper, this is a weather data set. And you can see we have the same problem as before, which is we have day 1, day 2, day 3, et cetera, et cetera, et cetera. When you get used to processing data, you'll start to identify like, wait, the first step I need to do is pivot longer. I want to say like 80% of the time, that's probably the thing to clean up your data set, is pivoting it longer. And then this data set, for its data for some ID, year, month, there, and a given day, there is a max temperature value and a minimum temperature value. Okay, so the first thing that we will do is we notice that our columns contain values and they're not variables. So we'll pivot longer. So again, we'll pivot our data set. That's the first thing that we'll do. And we'll have the value, the data set on our left. Okay, so you usually don't know you have this problem of variables being stored in both rows and columns until you end up in this current scenario that's in the left, right? So right now we have, for each row, it is for given ID date, we have a value, a temperature value, and then that temperature value is either a maximum value or a minimum value. But if we think about like what should an actual row represent, it should represent like the date level, the temperature of a date at a particular date. So we actually want the table on the right, which is we want a column that tells us the max temp and a column that's the minimum temp, and then each row just gives us like for a given day what's the range of temperature. The symptom that you have, so this is the scenario where you have data being stored in variables being stored in the row, right? We want a variable that's Tmax and Tmin, but it's stored in a single column and not separated out into two columns. So one way you can think about it is this is the total opposite way of pivoting longer, which is going to be called pivot wider. But the symptom that you see when you have a data set is if you ever end up in a data set where a bunch of information is repeated in a row, for example, ID, date, like they're all in duplicates. Like if you see like all of your demographic information like duplicated and the only thing that's changing is like a value or two, then that is a pretty good symptom that you have this problem that you need to pivot out a column to solve this data problem. And so this is what we'll do in R. So we will load up another data set called CMS. So this is the CMS utilization rates by state, demographics, year, and measurement. CSV. So we will read a CSV file. It's going to be under data, and it's going to be called CMS_utilization. And in our current data set, we have for a given state, a state FIPS code. So this is something when you work with like census data or any geographic data in the United States, you'll be having FIPS codes. You have a variable, so per capita annual spending in dollars for males aged less than 65 years, 0 to 1 chronic values, and the CMS utilization is 1,593.9423, right, for the year 2007. So this is the utilization rates for a bunch of demographics, and then I believe it is a dollar amount. Okay, so if we keep scrolling in our data set, we see that we have this problem where year is stored in columns, right? And again, when you're putting together your own data set, this is totally fine. I'm not saying that every time you start up a data set, you create the data set in a tidy format. That is not the case. Again, remember at the very beginning when I talked about like some data sets are easier to enter data and visually inspect, and then other formats of data are what a computer needs to make calculations easier. So the first problem we have is exactly the same problem that we've been working with this entire time. So we can say CMS_long is going to take the CMS variable or the data set, or its percent greater than percent. We're going to say pivot longer. And again, we want to select a column the columns that represent the columns that we want to pivot. So I'm going to say I want the columns using backticks, 2007 all the way to the last column, right? So that is selecting 2007 all the way to 2017 without me having to type each one of those out. Next, I can say names underscore 2. I'm going to turn those columns into the value year. It is last, L-A-S-T, last column. Okay, so now if I look at CMS long, you can see that we now have 8 columns versus before we had 17 columns. And now we have 38,000 rows versus before we had 3,400 somewhat rows. So it did that pivoting step for us. Okay, so now let's look at this CMS long data set. You can see that we have this column called variable, and we have a value for per capita spending actual. And if we scroll down a little bit, there's per capita spending standardized for the same state, male, sex, age group, etc., right? So we have a bunch of different, this end value at the end can represent ED visits per 1,000 beneficiaries, or it's the per capita spending actual or standardized, etc. So this variable column should be pivoted out such that we get something that's for given state, sex, age group, number of chronic conditions, and year, like what that calculation is. And so this is where we have to do that last pivoting step, right? So we essentially have this version on the left, and we want to pick out a column and turn each individual unique value in that column into a separate column, right? So in the element column originally, we have a Tmax value and a Tmin value. Those are the 2 unique values, and those 2 unique values become 2 columns. In our case, I believe we have like 5 unique values in this variable column, so we'll have 5 more columns at the end. So the way we do this, instead of pivot longer, we can say our CMS long data goes into a function called pivot wider. And pivot wider, because it only works with 2 columns, right? We don't need to specify a selection method. The only things that we need to provide is a parameter called names from. So what is the names? What is the column we want to pivot out? It's going to be this variable column. So we can say names from equals variable. And then we can say values underscore from. When it does this pivoting, what is the column that you want to use to fill in the values? And that's this last column called value. And so now we can save this into a variable called CMS underscore tidy. And I will look at it here. Oh, actually, let me paste this into chat. And now I can see for the state of Alabama, for males aged less than 65 with 0 to 1 chronic conditions in the year 2007, we have 4 different values, one for per capita spending, standardized spending, ED visits per 1,000 beneficiaries, and hospital readmission rates. Right? So this row is actually more meaningful than what we had before, where if we wanted to get hospital readmissions rate, we would have to filter our data set where the value is equal to hospital readmissions rate and then figure out what is going on. Now that the values per row is actually more meaningful. Okay, so that's really like the core fundamental thing about tidy data, which is data can be messed up in multiple ways, but we have a core set of tools mainly around pivot wider and pivot longer, and you can do a bunch of other things as well. But then at the end of the day, you have a tidy, cleaned up data set. And why did I spend so much time talking about this is because when you are trying to plot in R or in any language really, when you are trying to fit models, it takes a tidy data set to do those things. Also, if you want to combine data sets from one data set to another, it needs to be in this tidy format so you can do this combining of data sets. And so this is actually the most important skill to know, which is how to tidy up your data. And so as long as you are mindful when you're curating your own data sets, and because you've learned how to pivot longer and wider, when you are curating your own data set, think about what breadcrumbs you have left yourself so you can do these transformations a little bit easier, or give the analyst like some hope that they can tease out the little components of you manually entering your data. Right, so a little bit of forethought in putting together your data set can save a lot of headache later on when you're doing the processing. Okay, so let us take a 5-minute break. So come back at 50 to the hour. And I will put up a timer. And if you have any questions, just post them in chat somewhere. You can either DM me somewhere. But let's take 5 minutes, come back at 50 to the hour, and we will cover visualization, and then cover an introduction to visualization and an introduction to fitting a model. Okay, welcome back. So I found out. So if you haven't noticed, or if you're having issues with your R installation in the main website, there under the setup, there's a section called Binder, and under Binder, if you want to like practice this in like the university library, or like you're on another person's computer and you don't have R, they don't have R set up, you can literally click this first button to open up a Cloud RStudio session with our data set and packages installed. Having said that, there's one of you who couldn't get RStudio installed because of university issues, and the pivot longer, pivot wider functions don't work. And I just found out that is because the package version that is installed in the Binder instance is older than when they use pivot longer and wider. So if you go back into like the actual notes, you'll see that the words gather and separate are being used. And so this version that I just realized is actually using the versions, instead of pivot longer, it is the function called gather. And instead of the function pivot wider, it is the function called spread. So it's instead of longer wider, it's gather and spread. Gather and spread was not an intuitive name, so they renamed everything to pivot longer and pivot wider. But having said that, I will update the version of R in the packages in the instance, so it is a proper like newer version of it. So that is the issue that was going on behind the scenes. Okay. So the next thing that we're going to talk about is visualization, and there is a lot you can talk about visualization. You can have an entire course about this across 13 weeks and 3 hours a week, but we're going to do this in about 30 minutes. So we're going to introduce you to how ggplot works, and if this topic is something that you want more of, like think in the terms of like 1 or 2 more hours, then let me know in the feedback. So when we build the part 2 session, we can either do more on visualization, more on actual like real world data processing and cleaning, or we can do more on fitting models. But usually fitting models is its own 4 to 8 hour day. So do let me know. But we will introduce ggplot in the next half hour. So the most important thing that we want to know is there is this notion of different types of information variables can store. So we can have nominal variables, which is discrete things that are not related to one another. So, treatment group 1 and treatment group 2 are not really related to one another, even though they have the values 1 and 2. You can have, control and experiment, right? So those are nominal values. You can have ordinal values like very commonly like Likert scales, which are like select which one do you agree with, like neutral, like disagree, strongly agree. Those are on some numeric scale, but you don't have a value of like 1.5 or 1.572, right? They're in discrete steps, but there is an inherent order to them. And then some combination of a very specific version of a nominal value is a binary value. And so this is really common when we're working with health data sets, because usually our outcome of interest is a binary outcome, right? Did this person get cancer from smoking? Yes or no? Did this person get treated, get better from this treatment? Yes or no? Like, if you can ask your question in a yes or no format, that is a binary variable. Yeah. So speaking of, so this plays into a role in visualization because the type of data that you store will dictate the type of visualization you can make. And so in ggplot, this is a slide from Thomas Lynn Peterson. He's the current maintainer of ggplot, and he works at RStudio. Some side backstory, ggplot, the R package, was actually Hadley Wickham's dissertation project back when he was at the University of, I forgot where he graduated, so I'm not even going to try to make that statement. But so ggplot stands for the Grammar of Graphics. It's based off of a book called The Grammar of Graphics, and during ggplot and ggplot2 is the implementation or the code implementation of that grammar of graphics. So what is this grammar that we are talking about? So in ggplot, the way we specify how a plot gets created is in a single plot's constituent parts. So it is a layering system where we have a data layer at the bottom. Then we specify some kind of aesthetic mapping. So in our previous example, we had like our CMS data. That is our data value layer, right? So now if we talk about anything, it's using that data frame. The mapping, the aesthetic mapping, is if we have a plot has a x-axis or a y-axis, which column corresponds to which feature of our data set, right? So maybe you want x to be one variable, y to be another variable, the color to be another one, the shape to be another one. Those are all part of the aesthetic mapping. On top of that, certain plots like bar plots inherently do a statistics calculation, right? So if you create a bar plot off of a single column, by default, the statistic that is being calculated is the sum, right? Because bar plots measure the count of something. So you have a statistics layer. Then you can have a scale layer. So certain blood values are going to be measured on a logarithmic scale. So now you can specify, do you want the x-axis to be on a log scale or not? Do you want it to be on a base 10 scale or not? So then you can specify scales. Up until now, we haven't actually drawn anything to the plot. So then you have a geometry layer which says, what do you want drawn on our figure? Do you want points? Do you want bars? Do you want a histogram, which are really like lines? Do you want a box plot? Those are all geometries. On top of that, we can add facets, which I'll show you. It's easier to show. And then you can change the coordinates. So this is the coordinate structure. So we're used to something called the Euclidean coordinate, Euclidean coordinates, which is, 1 unit in a box. But you can have polar coordinates, which you draw your figure in a circle. And the very last thing are the themes. Do you want your x-axis rotated? Do you want your font for this to be larger? Do you want to get rid of this background color? Those are all theme elements. And so this is how ggplot works. So you specify every single one of those layers. And the nice thing is, during this entire stacking process, if you don't specify something, it'll pick up the data from a bottom layer. So you don't, even though like the geometry or the aesthetic mapping is tied to a piece of data, you don't have to resupply the data set every single layer because it'll just drop and use the base one if it can't find it. So that is how ggplot works. And that is not the 30 minutes. We're actually going to write some code. So I'm going to open a new file. And we are super pressed for time. So 04, and I'll call this one ggplot. or ggplot2. That's what I will call it. So this is going to be a new file. So I'm going to go to session and then hit restart R. So all of my data sets on the top right corner are deleted. All of my packages are deleted. So there's two things I want to show you guys. One is we have to reload tidyverse. The second thing is we want to load up the ggplot2 library. And the third, we're going to actually use data from the medical data library. And I actually forgot to have this in my notes. Just give me 1 second. So there's to install the medical data set. It's a little bit more complicated than installing tidyverse from our original instructions. We actually need to install a package called remotes first, and then from remotes we install the medical data set that Peter has given us, right? So Peter's repository with all of his data sets, with his code books and descriptions are all on GitHub. And so we want to install this package straight from GitHub. So if you run the first command that installs remotes, and it's going to say, do you want to start your R before reinstalling? Hit yes. And it's going to go through the process of installing the remotes library. And so I'm doing it right now. Once you're done, I pasted this second command into chat. And then this is Peter's username, and this is the package that he has on GitHub. So now I can say install this package from GitHub, right? I already have it installed, so it's going to say like, hey, I'm not going to do this again because you already installed it, but that is how you have that install things from GitHub. And so if you need like a super updated version of an R package that's on GitHub, this is how you do it. So just give me a green thumbs up, green yes, when you are ready for me to continue. Or a red check mark when you like are absolutely lost, but okay. So I will move on to the next part. So because we went through this installation process and restarted R, we have to load up tidyverse, load up ggplot, and now we should be able to say library medical data. And now we have access to all of Peter's medical data sets. First thing we're going to do is use one of his data sets. And instead of typing the full name, there's a data set called blood storage, but we're not going to want to type blood_storage every single time. So I'm going to say, give me the value blood, and then assign it the data set blood storage, right? And this will only exist if you load up the medical data set, right? So one really useful thing about R is you can actually submit data sets into R packages. So if you're trying to create like a reproducible study, you can literally make an R package of all of the data, the raw data that you use for your study, and then distribute it. So they don't have to go and look for a CSV file or an Excel file somewhere. It's actually packaged right into R, which is really nice for teaching examples, because then I don't have to show you how to load up a data set. I can just use it straight up. So one thing, if I want to look at this data set, I am simply in my console typing in the data set and hitting enter and just running it on its own. In this current example, if we just type in blood, you can see that the printout is a little bit different from the data sets that we used before. And if we look at the function class, so this was something that I believe we covered on Tuesday, when we looked at the class of a data set and it told us it was like a TIBBLE/TIBBLE-DF data frame. This blood data frame is a regular, pure base R data frame. That's why we don't get a nicer, prettier printout and we get everything dumped to our screen. But again, because this is a data frame, we can do regular data frame things to it, right? Okay, so now let's actually create a plot. Remember the layering system with ggplot? So we're going to be using the first layer, which is ggplot. Then we can say we're going to specify data layer, and our data layer is going to be this blood data frame. We hit enter, and then on the right panel, it should open up a plot. And it should be a blank plot because all we have done in our layering system is specify a data set. We haven't done anything to it yet. So next, if we in ggplot hit comma and hit tab, you can see the next thing to supply is a mapping. So remember the layering system, we supply an aesthetic mapping. So mapping equals, sort of one of the weird quirks with ggplot, you have to say mapping is equal to an aesthetic. An aesthetic is the function called AES. So we can say mapping equals AES. And in here we can say, what do we want on the x-axis? This data set is about, what is this data set about? This is a data set about 316 men who have undergone radical prostatectomy and received transfusion during or within 30 days of a surgical procedure and had an available PSA follow-up data. And the outcome of interest was time to a biochemical cancer recurrence. So we're curious about whether or not someone had a biochemical cancer recurrence. And that variable is stored in a variable called recurrence. So we can say x is equal to recurrence, R-E-C-U-R-R-E-N-C-E with a capital R. And now if we hit enter, again, we told it to plot on the x-axis the recurrence variable, but again, if we think about the layering system, we actually haven't told it any geometric layer, right? Like, what do you want plotted? So now, on our next layer, how do we add up to the next geometric layer? This is all the data layer information. The next layer, we can say plus, so we're literally adding another layer. All of the geometric layers begin with GEOM, geom underscore. And here, if we pause for a little bit, RStudio will literally pop up all of the different geometric layers that we can provide. You can have an error bar, you can have a curve, you can have a bar plot, so geom bar, histograms, violin plots, box plots, etc., etc. So what we want is a bar plot. So we can say geom underscore bar and hit enter. And then finally, because there's enough information to create a plot, ggplot created a plot for us, right? And a bar plot, the default statistic is to count that information. So now we have a count of the values that are 0, which is somewhere in the 200s, and the values that equal to 1, which is about 50. So it's really important when you're looking at visualizations, when you're exploring your data set, that you look at your variable of interest, right? So what is the variable of interest? This is sometimes known as your dependent variable. I actually do not like that term. Or sometimes you might hear this called, what is your Y variable? So this is the outcome of interest. Sometimes it's called your outcome variable. And in the statistics / machine learning world, you'll hear this being called the response variable. So you'll hear, and then the predictors are either going to be your X variables, your predictor variables, sometimes called your independent variables. Those are all synonyms for the same thing depending on what field you're coming from. In the machine learning world / stats world, you'll hear predictor response the most often, or X and Y the most often. And then independent, dependent are usually terms that are used in the social sciences. Okay, so just for the sake of time, there's another thing. Remember when I said we can swap different layers, and if a layer doesn't exist, it'll drop down to the previous layer, right? So we can actually copy this last line and say mapping equals AES. We can get rid of that, and don't forget to get rid of that little trailing comma under ggplot. We can put that mapping layer into the geom bar layer, and we will literally get the same information, right? So it's going to say, hey, you're going to plot a bar graph. You're going to tell me I'm going to use X, the recurrence variable for X, and I'm going to do that, but I don't know what data value is. So I'm going to drop back to the previous layer and use the blood data frame from there. And we can take this one step further, and we can actually start off with a completely empty ggplot and specify our data in our mapping in the last layer as well. And we get the same exact plot. These things are all equivalent to one another. The reason why this is beneficial is you can actually plot two different bits of information from two different data sets into a same figure. So this is what it gives you. You can specify a different overriding data layer if that's what you need. Okay. So now let's just explore some of the ggplot, explore a little bit more with ggplot, and let's pick a figure that uses two variables. So we can say ggplot, data is equal to blood, and I'm going to add another plot. I'm going to say geom_boxplot. So a box plot, the way it works, so we can say AES for the aesthetic mapping. We can, on a box plot, in the box plot, typically what you have is you get a separate box per categorical variable, right? So let's say X is going to be the tumor volume variable, which represents the tumor volume as an ordinal variable. So either the tumor was low, medium, or extensive. And then we can say on the Y, give me a continuous variable, let's say age. And so we get a single box plot, but here's the thing with this value is tumor volume in our data set is encoded as 0, 1, and 2. But those are ordinal variables, and because it's written as 0, 1, 2, R is treating it as a numeric column. So just like before when I said as dot numeric to convert a string into a number, we can actually tell R, hey, I know that you are given like a number or a string or something, but I want you to actually treat this as a categorical variable. And in R, a categorical variable is called a factor. So we can say as.factor of this tumor volume. And so now it knows, okay, tumor volume is given 0, 1, 2, and 3, but you want me to treat it as a categorical value. And so now if I run this, I now get a separate a box plot for each unique value of tumor volume. Because I've told it, hey, treat this thing not as a number, but treat it as a categorical value. So I personally, when I'm drawing box plots, I always draw another plot called a violin plot. And so what does a violin plot look like? We can say ggplot, data is blood. We're going to add, and after the plus sign, you can create a new line. So you can sort of, just like the piping syntax, it's a little bit easier to figure out where your layers are. I'm going to add a geom called violin. And I'm going to say AES, X is equal to as.factor tumor volume, T volume, and Y is equal to age with a capital A. And this will draw what is known as a violin plot. The way you read this is essentially think about cutting the violin in half, and then on one half you have a density plot. And why I like violin plots over box plots is because you're able to actually see the distribution of your data much better in a violin plot if it's skewed than in a box plot. So if your violin plot is more or less normal, like with a classic hump in the middle, then you can get away with using a box plot. But as soon as it gets skewed, it's a little bit harder to see on a box plot that it's skewed by, sometimes you can have extreme skews and it doesn't show up as like, it's not as big of an extreme when you're looking at it as in a box plot. So one other thing you can do, remember when I talked about adding different layers, there's a layer called jitter, which essentially says, I'm going to plot the points, but I'm going to on purpose plot the points in a random position just so they don't overlap on one another. So we can say add a jitter layer where x is equal to as dot factor tumor volume, y is equal to age. And so when I talked about adding multiple layers on top of one another, this is using a violin plot as a geom on the bottom, and then a jitter plot as another geom on top. And this order does matter. So if you put the plots at the points first and then you put the violin, you're actually covered the points. So this layering system in ggplot is super important in terms of the order. Okay, so last thing is how do we deal with multiple categorical values? I'm sort of just for time going to end up going into copy paste mode. And so I'll paste in the commands that I'm running. We can put a scatter plot, plotting the volume on the X and the pre-op PSA on the Y. And we have the same issue with the Y where the pre-op, and we can also, as another aesthetic layer, pick another column to represent a color. And so all we need to specify is color equals, and then the name of the column. And this is also a categorical value. And so you can see it gave us like a continuous scale, even though it is a categorical scale. So we can say as dot factor on this. And now we get for each unique value of SGS, a different color point in a scatter plot. And so we can keep building on this to the next point, which is called a facet. A facet, what this allows you to do, you can see we have an overplotting problem right now. In this bottom left-hand corner, there's a lot of points one on top of each other, and it's hard to tell what exactly is going on because they're literally all plotted on top of one another. So we can actually, just like the group by, where we like created a separate calculation for each group, we can create a facet, which is a separate figure for each group. So we can say, for example, facet wrap tilde. This is sort of the weird quirky syntax that you have to get used to, but we can specify another categorical variable. So rbc.age.group, and I will paste this into chat. And what this will do is for each value of RBC age group, so each value of age group, it is going to replot a scatter plot with the SGS as a color. And so this is a really good way to start parsing out how variables are related to one another in your data set. So I'm going to copy that last line one more time, and then instead of facet wrap, we can specify two variables using a function called facet grid. Here, we still use the tilde, but you can specify a tilde to the left or to the right. So one variable to the left of the tilde, one variable to the right of the tilde. And then here we can say that we now get the faceting based off of two variables. It's the same scatter plot and color, but now we have a separate figure across the top. It is going to be the age group, and then across the rows, it's going to be the reoccurrence value. And so when you're trying to see like, hey, of the people that have reoccurrence values, like you can maybe see like, hey, maybe like these points are a little bit different from the other points, and that's probably a variable that I want to get into for my analysis. And the last thing that I will just show is how do you sort of clean up the colors or figure or text and all of that stuff. There is a last layer called theme underscore whatever, and you can say theme underscore, I think minimal is the one that I use the most, and it can get rid of a lot of the extra stuff in your figure. And this is to say theme underscore is pre-made themes, but if you're working at an institution that has like its own color palettes and color scheme, you can create your own theme and then apply them to your figure. So maybe you need like all your fonts to be a certain font. Maybe you want without any grid lines. Those are all different theme values. Even stuff like rotating x-axes and stuff like that. So that's all I want to cover in that case. Okay, so we are super short on time, but I want to be able to show you, instead of writing the R code, I will talk you through the R code. How's that? The last topic is around fitting a model. And the model that I have in my notes is actually a logistic regression model. And why I picked logistic regression was mainly when I was doing my master's in epidemiology. That was probably the most used model that we were taught and used. And so a logistic regression model is a model where the outcome is a binary outcome. So we care about this in health because it's yes or no. Did this person get cancer? Yes or no. Did this person die? Yes or no. Did this person get better? Yes or no. As long as we can answer that question as in a yes or no binary format, we can use logistic regression as the model to use. And so, again, remember, logistic regression only works if your outcome variable is binary. It doesn't matter what your other predictor variables are. It is only your outcome or your response variable that matters. If the variable you care about is binary, then you can use a logistic regression model. Logistic regression is essentially a straight line. So remember from y equals mx plus b, that is the formula for a line. The formula for a regular linear regression model is y equals mx plus b, but every mx piece is a separate variable. So we can have a variable for age, a variable for tumor volume, pre-op PSA, et cetera, et cetera. Each one of these variables is going to be called, another way they're referred to is a beta. Those are going to be parts of our model. And so linear regression on the left is drawing a straight line. Logistic regression, because the outcome only is 0 or 1, we cannot just draw a straight line because that would be a terrible model because a straight line will go into negative infinity and positive infinity. But our logistic regression model is literally only 0 or only 1. So this is where picking a model becomes really important because if you draw a straight line, yes, you drew a straight line, but it is completely the wrong model because it doesn't fit in the parameters of the data set you're working with. So how do we go from straight line into this S-curve? There is something called a logit function, which is log of the odds. So you can think of odds as the probability of something happening. So the probability of flipping heads is like 1 over 2, where 1 is heads over 2 possible outcomes. The odds, instead of 1 over the total number of outcomes, because in the real world we don't know what that denominator is, we say is the probability over 1 minus the probability, which is also known as the odds. So that's why when you work with logistic regression models, if you've read about them before, you talk about odds ratios or ORs, and that all comes from a logistic regression model. And so essentially the thought is, to go from a straight line to this S-curve line, we applied this function. It's called a logit function, but we apply some function. And then when we want to interpret our results, we undo that function, and then we are essentially back to drawing a straight line. So in general, logistic regression is drawing a straight line that we do a special function on top of. What does this look like in R? And we can load up our blood storage just like before. I will make this huge. And we can run the GLM function in R to fit a logistic regression model. And the way models are fit is you specify a formula. There's a tilde involved. To the left of the tilde, that is the variable you care about. So this is your y. So remember y equals mx plus b. This is your y. So this is y equals on age. You specify the data. And then to make it logistic regression, you say family equals binomial. That's the only thing you need to remember in that case. You end up with a model object. And then here, you'll end up with a coefficient. So you can see that for age, the coefficient is 0.015. And remember, you can run this function called summary to give you the actual full statistics output. Part of it is this coefficients table with the p-value that you can interpret. So the problem is, the way we interpret this is for every 1 unit increase in age, the log odds of being positive, having a positive indicator for our blood is going to increase by 0.015 times. So that's a really weird like interpretation because we have to call it the log odds of something. Remember that logit function is log odds. So we can undo that function by saying, instead of the opposite of a logarithm is the exponentiation function. So we can take that model and exponentiate that column, and now we can have the odds ratio, in which case we can now say for every 1 unit increase in age, the odds of, I literally wrote it down, the odds of earlier biochemical reoccurrence of prostate cancer after prostatectomy increases by 1.02 times for every unit increase in age. So that is the only way you can interpret that number. It is a very technical sentence, and that's essentially how that works. So there's only one other trick that was involved. There's a package called broom, and if you give broom, there's a function within broom called tidy, and this is to not be confused with tidying data sets, but there's a function called tidy in the broom library. If you give it a model, what it will do is take all of that weird stats output and give you a single data frame object back. And so that's why when I had this data frame object, I can then run this mutate command and assign a new column called odds. That is the exponentiation of this this estimate column to get this odds ratio value for our logistic regression model. So I just want to stop there because there's like literally just 4 more minutes. I will say this whole write-up is written in the analysis chapter. And so, but the main takeaway from today was really understanding tidy data, because if your data set was tidy, you can see the way we specified this model is adding another column. So if you cared about, year as a column, as a variable, as an input for your model, you need year to be a column and not 2007, 2008, 2009, et cetera, et cetera. And so this all ties back into like tidy. A lot of tools rely on your data set to be tidy. And so that is why the vast majority of our time was spent on teaching you tidy data, because that feeds into plotting and it feeds into models. And so with that, we will sort of just go through the concluding slides. So hopefully this was sort of a somewhat gentle introduction to learning about R and programming, and hopefully it wasn't as scary as you thought it could be. I mentioned on Tuesday that there is a huge community that is out there wanting to help you. And so there is this book called R for DS, which is the data science, everything about data science version, written by Hadley and Garrett Grolemund. That is also free to view online and has a very similar format to the materials that I have. There is the R for Data Science community, which you can join their Slack group if you need people to ask questions for. That is always the hard part, is you're learning a new skill and you need to ask questions to learn. So there is a Slack group for the R for Data Science community, and it's run by John. I just know his last name is H, starts with the letter H, not our John Briganti, but another guy named John. What's also cool about the R for Data Science community, there is literally weekly office hours. So if you do have a problem with your R code, quite literally hop into office hours, and they have a set of TAs to sort of answer our questions. Another thing, there's a thing on Twitter called Tidy Tuesday. The best way to learn this R material is to practice this, and what ends up happening is every Tuesday, you can see this on the hashtag Tidy Tuesday on Twitter, a data set gets released, And you essentially just, the task is work on this data set for 1 hour to create something, either if it's a summary table or it's a figure. Just work on this data set for 1 hour, and then people post their results on Twitter, and they sort of, you see all the results come in throughout the week. Speaking of Tidy Tuesday, David Robinson, I spelled his name wrong, it's David Robinson, streams his Tidy Tuesday stuff every week on Twitter. So if you just want to see more of the Tidyverse set of tools being used and what can be used, or ask him questions, or hear his thought process, you can watch him on YouTube every Tuesday, where he literally takes the data set he's never seen before and processes it into a data set, into a visualization. If you are part of a minority group, or underrepresented group, or if you identify as being female, there is the Our Ladies Global Slack channel, which is another safe place for you to ask questions. And then there is also the Our Medicine community, and if you want to join that Slack channel, just message me and I can send you the Slack invite for that, which is, these are literally only like the medical doctors and nurses. We throw a conference every year talking about R and how it pertains to the medical field. So yeah, with that, feel free to contact us. Data Services at University Library has consultants that can help you work with dealing with your data set. You can contact me or Anne around that. The Alex Hanlon runs the Center of Biostatistics and Health Data Science here in Roanoke. She also so happens to be on my committee, and she does all of the work with helping people take their health data set and do some type of project analysis with it. So in one side, you have me and Anne and university libraries helping you process data, and then Alex will help you do the actual stats analysis. So that's sort of how that interaction works. And you can, and what's also really cool is her group literally has drop-in Zoom consulting hours on Monday and Wednesday for a very large block of time. So I use it myself as well, just to ask them about stats questions. So you can as well. And then last but not least, the Carpentries. Nathaniel Porter is the head of the main contact for the Carpentries at Virginia Tech. All of the materials, how I learned to teach, and a lot of my teaching style is based off of the Carpentries. And so you can, if you want to learn about Python or more about R or other things, other community lessons, you can check out any of the Carpentries materials as well. And then last but not least, if you can, please give us feedback about how this went. I am currently 1 minute over time, so I will just leave this slide up and just say thanks for attending the 2 days, and hopefully you have learned something, and hopefully R has become a little bit less scary. And remember, from expectations from the very first day, I do not expect you to rewrite your R program tomorrow, but if all you did was load R for a data set, spend 5 minutes, 10 minutes, that's all you need, load it in. Even if you don't do anything, just getting used to the interface is how you can slowly progress and start incorporating all of the tools and techniques. And with that, I would say thank you. You are free to go. And if you have any concluding remarks, you can make them as well.
Yeah, myself and Dan will stick around for a little bit. If you have any individual questions, we can help you with that. Be on the lookout. We'll send out additional workshops that we're doing. If there's other questions and you want to have one-on-one sessions, we can certainly facilitate that. So we thank you for being here, and good luck exploring R.